Nina Tandon, an American biomedical engineer and the founder of the groundbreaking skeletal reproducing company EpiBone, was born on May 8, 1980, in Roosevelt Island, New York. Although she may participate religiously in meditation and loves to run marathons, Tandon has dedicated her life to science wholeheartedly. She originally became interested in science growing up because her siblings suffered from genetic eye conditions, and she was always curious if there were ways that she could help them. Following her high school journey, Tandon began her educational journey at Copper Union, graduating with a bachelor's in electrical engineering in 2001. Then, from 2003 to 2004, she attended the University of Boom Tor Vergata, where she worked on a device called the Libranos. Tandon went on to graduate from MIT with a master's in electrical engineering from 2006, and then studied at Columbia, graduating in 2009 with a PhD in biomedical engineering, with a focus in cardiac tissue engineering. While at Columbia, she worked on creating human tissues and got her MBA there in 2012 with the intention of being able to make the results of her research more available to people who could benefit from them. Tandon now studies biomedical engineering, and her field of focus has been primarily in the creation of human tissues from stem cells. She spent her whole career, including her time in schooling, exploring the matter of how feasible it would be to regrow damaged bodily tissues, such as bones, hearts, and other vital organs, utilizing living organisms. Additionally, she makes that question even more complex because she also explores the feasibility of making procedures to regrow these important tissues a quick and inexpensive process for both clinics and patients. In 2019, Nina participated in a study on the imaging of stem cells within the ovarian tissues of flies, called germarium. The research was designed to create a more efficient and non-damaging way to observe the stem cells within a commonly studied organism. Tandon's team worked to fine-tune a specialized silicone imaging chamber that would minimize the movement of the germarium, as well as the damage taken by the germarium during the experiment. They first created a reagent and built the imaging chambers. Then they raised the flies in the experiment to the same age, and then they surgically removed the ovaries from multiple flies. After that, they removed the germarium from the ovaries and separated them for imaging, injecting each one with a green fluorescent pigment. Then finally, they took images of the germarium. The figure shown on screen represents the images they got during their experiment using their fine-tuned silicone imaging chamber. Overall, the figure is just meant to show generally an inside look of the stem cells in flies and how those stem cells may move or change over time. Section A of the figure displays the images of the green fluorescently labeled germarium as they moved over time. Section B represents a 2D diagram of the movement of those cells over time. Section C shows the relative movement of the cells away from each other, the positive values listed in blue, or toward each other, the negative values listed in pink over time. And lastly, the sections D display images of different damaged germarium that they deemed not to be important to the experiment or relevant. Tandon's work with stem cells and flies likely helped largely as an inspiration for her current passion project. Tandon started her company EpiBone to work on continuing exploring the exploration of possibilities of stem cell tissue reproduction. After her many years of working with the reproduction of various working tissues, she used her knowledge to fine-tune a process for the reproduction of tissues within living beings. The process of recreating a bone for her is simple. First, get an accurate CT scan of the damaged tissue. Next, harvest stem cells from the patient's fat. Then, place the cells in an incubator where they grow to the size and shape of the damaged tissue. And finally, surgically place the bone back into the patient. Tandon has gotten wide recognition for her personalized approach to tissue reconstruction, and her company is in the works of getting the process approved for use in surgical settings. However, although Tandon generally grappled with few large hoops on her way to the company's development, getting to the place where she truly wants to be with EpiBone may prove to be challenging. For one, Tandon has only tested her process on animals and not humans, thus making it hard for the FDA to approve her process as legitimate and safe. Additionally, the process is incredibly costly and would be hard for patients to choose over traditional bone transplants, which are roughly a fourth of the cost. Although these hurdles may be difficult to overcome, Tandon is working hard at making her ideas and dreams possible. I really admire that Nina has dedicated her life to creating medical solutions utilizing pieces that already exist inside the human body rather than with chemicals and medications. even when the process of getting there is very tedious and challenging. I'm sure as she continues on her journey as both a scientist and entrepreneur, she will be able to do some more incredible things for the world of science.